Hi, my name is Drausin Wulsin. I'm the ML lead here at Immunai. I'm excited to talk with you today about reprogramming immunity with artificial intelligence and single-cell multiomics. So today I'm going to talk about how can we engineer our immune system to be more effective. Part of this involves AI problems and what problems do we need to solve in order to do that. I'm going to then talk about how solving the lower level problems allows us to start thinking about answering the higher level ones. I'm then going to talk about how solving the higher level problems allows us to design better immune system soldiers, aka T cells. And finally, what is the interaction between artificial intelligence and in vitro cell therapy experiments as we're optimizing these immune soldiers? So our immune system is better than any drug we could possibly build, especially when we're fighting zombies, aka cancer cells. But as we know, it's not always enough. And our goal at Immunai is to use our understanding of core immune biology to build better therapies. This means engineering T cells. Genes are the source code of these cells, and so we optimize T cells by modifying their gene expression. How do we do this? We take blood samples from cancer patients, and we put them through single cell analysis, which means identifying and studying each individual cell using these technologies. For each cell, we have this amazingly rich set of data. We have gene expression, we've got surface proteins, we've got TCR and BCR receptors. These are receptors on the surface of the cell, of the T cell and B cell that help recognize foreign invaders. And we take these insights at the clinical level, for example, looking at what patients respond, what attributes of patients who respond versus don't respond are there. And we use it to guide how we want to genetically engineer better T cells. So the challenge in this enterprise is learning enough core biology about the immune system so that when we study, the previous N diseases, it gives us a major head start when we want to start the N+1. Right. So the fundamental assumption here is that the immune system has many similar attributes in how it behaves in arthritis, irritable bowel disease, cancer, infectious disease, psoriasis, as examples, which allows us to understand how it's behaving in, let's say, diabetes, without having to start from scratch again. So at Immunai, we approach these problems first and foremost with lots of data, right? And so the data we have mostly falls into two different categories. We have what are called observational clinical samples. Think of these as sort of vials of blood, although we also do work with tissue as well. And we have in vitro perturbation experiments, which is basically data coming from experiments we do in the lab, where we're perturbing the individual cells through gene editing. In order to understand all this data, we use lots of AI, right? So right now, Immunai is only about 2 years old. We're solving about 10 distinct modeling problems in production. By the end of this year, I think we'll probably be solving another 10 more, and it's only going to increase. In order to make sense of this data and to use all this AI, we need lots of multidisciplinary expertise. This is everything from immunology, single cell science, thinking about how to actually process and analyze all this data through our infrastructure, using computational biology, and of course, artificial intelligence. So in order to do this, we have to solve many biological problems using AI at varying resolutions, right? So we start at the cell level problems. So cell level problems are things like noise removal, and I'll talk a little bit about that in a moment. things like cell type labeling. What kind of immune cell is this? Is this a T cell or a B cell? Or more importantly, what T cell subtype is it? At the sample level, we have things like technical batch correction and cell type abundance. At the gene pathway, so gene-gene interactions, we have to infer, are there new novel pathways that haven't been studied before that we need to find? For an existing pathway, how do we understand the causal relationships between the genes within that pathway? At the patient level, we have things like disease characterization. Is this patient going to respond well to this disease or not? And we have, how is the patient's immune system behaving in the face of a particular disease, whether it's a tumor or something else? And finally, at the disease level, we want to be able to think about characterizing the patients who tend to do well versus not do well for under a particular treatment, and using that characterization to identify new targets for gene therapy and for therapies. So this all starts with data, and our data is stored in a database called Amica, right? It's our centralized database of single cell analysis, and it is, if not the largest, and certainly one of the largest centralized databases. We have over 500 patients in it, 20 diseases, 15 plus treatments, about 3 time points per patient, which is a ton. And as I was going and looking into running some queries against our database to see how much it was growing month over month, I found this linear trend, which when you translate a linear trend into a cumulative trend, basically take the integral, we have a quadratic growth month over month. So what this means is that our database over the last year has grown quadratically, and it's going to continue to grow that way. And sort of just like nothing in the real world grows quadratically. And so I want to impress upon you just how exciting of a time it is. We are just getting so much more data about the immune system. And I think, and part of our central bet is that this data, this massive growth of data, is going to allow us to just understand it at a whole nother level than we've been able to thus far. So an example of a cell level problem is doublet detection, right? So doublets are a technical artifact of single cell genomics, where instead of a single, looking at a thing that you think is a single cell, you're looking at actually multiple cells, where we have multiple cells that sort of get encapsulated in the same bead, the same droplet. And this is a detail of how single cell genomics works. And if you have this and you can't detect these doublets, it really muddies the waters downstream. So it's really important to be able to detect when you have a doublet and to filter out those cells from your downstream analysis. But what we realized is that at Immunai, we had such a wonderful database that we were able to sort of use a pretty simple, pretty standard model, and coupled with our massive data, and just beat the pants off of the industry standard doublet detection algorithms out there. Right? So here we're comparing Immunai's doublet detection on the right to a standard method called Scrublet, which actually I think is a very clever. I like this method a lot. It's not a knock on the method. It's just the method was developed in a very sort of data poor environment, or it doesn't assume data rich environments. But for those of us who have data rich environments, we can do much, much better. And we can see that with our recall chart here, where Immunai is able to filter out far, far more doublets than Scrublet is. Another example of a cell level problem that we have to solve is cell type annotation. Right? And so consistently analyzing and characterizing individual cells allows us to be really specific in the subsets that we use downstream. And we'll see how that's impactful in a few slides, right? But historically across the industry, high quality annotations require this like really manual process by sort of very highly trained expert immunologists. It takes many days. Whereas Immunai, we have a machine learning driven process that's 3-tiered with the human experts only at the end, right? And so the core of this are supervised classifiers, right? These do a sort of an initial pass of cell type predictions and noise removal like doublets. We then have wrapped around that an expert system, which uses some additional domain knowledge to do some cell type annotation refinement. And more important, or almost more importantly, also adds a layer of quality control checks and auditing, right? And anyone who's used machine learning in a production environment knows the importance of QC and auditing trails. And then finally, on top of the expert system, we have the experts, the human review. And usually what they're doing is sort of reviewing the results that our automated process has generated, making sure that they make sense, and occasionally doing some minor adjustments for project-specific cell types that we care about. Right, and so if we think about what granularity of cell types can we detect, the industry standard granularity for immune cells looks something like this. We've got about 6 major cell types, and in each of those, we've got a few subtypes, right? And this is pretty nice. But what we found is that when we invested in this sort of 3-tiered approach at Immunai for annotating cells, we were just able to get way more granularity in terms of the specific cell types that we have. And that granularity and that consistency across annotations allows us to just have a much richer immune system mapping. So for a given project, we can just understand the landscape of what someone's immune system is doing in way higher resolution than we can with sort of industry standard annotation approaches. So how do we solve this on a modeling level? Well, we have a model called Hydra. And the sort of core insight here is that a lot of these cell level problems use the same multiomic data, right? So they use sample indicators and lab metadata, gene expression, protein, surface proteins, T cell receptors and B cell receptors. All of that goes into a few transfer learning layers. And then we use have some multitask learning where we actually have multiple cell level problems that we have to solve in order to sort of do our job well for subsequent analyses. And Hydra sort of manages to transfer the learnings. A lot of the learnings required to solve each one are shared. And so we can make use of these multiple problems into a single model. So solving these cell level problems allows us to begin thinking about disease level problems, right? And the one disease level problem that we were interested in solving is just treatment prediction. How well is someone going to respond to an immunotherapy if we give it to them? Right. So the challenge here is that the number of observations we have is limited to the number of observations in, let's say, an arm of a clinical trial. Right. So this is, let's say, at the most, we're talking about 50 patients usually for the data that we have. But the feature space is huge, right? So it's combinatorial in cell types, in genes, in features of genes, in other modalities as well. It's on the order of 10 million possible features, right? So really what this boils down to is a good old-fashioned feature selection problem, right? And we solve this through some thoughtful combination of domain knowledge, sparsity-inducing models, and lots of cross-validation. So when we started out doing this, we said, okay, let's just take the sort of standard approaches for cell type annotation and doublet removal. Let's see how well we can do. And, not that surprisingly, we weren't able to do that well, maybe just a little bit better than chance. Interestingly, when we swap in the standard annotations for Immunai annotations, our model gets actually much better. And the reason it gets better is because the features going into the model are cleaner. Because the features going into the model are for specific cell types. And so when you have better cell type and more higher resolution cell type identification, the features can be better. And when we combine Immunai annotations with Immunai's doublet removal, we're able to see that our prediction performance gets even better. And sort of the thing that surprised us is, going from accuracy in the low 60s to the mid 70s, sort of we were not expecting to be able to do as sort of respectably as the mid-70s for predicting whether a patient is going to respond to an immunotherapy. But I think the thing that was cool to see is that the difference in these, solving these fine-grained cell-level problems well is the difference between being able to do well on these sort of like higher-level disease problems or not. Right, but the models, these prediction models where we're predicting whether someone's going to respond, they're not interesting in and of themselves, right? What they do is they give us insight into some core immune biology, right? Because if a model can predict whether someone's going to respond or not respond, we believe that it understands something core about the immune biology. And what we can do is we can introspect that model to look at, for example, what genes are really important. What gene expression is it looking at in order to make its prediction? Right, and what we can do is then combine this gene importance from our model introspection with other analysis. In particular, we're thinking about biological pathway network inference, where given a particular biological pathway of, let's say, 100 genes, we want to learn the causal relationships between those genes, right? And with that network, combined with our signal on, let's say, the responder, non-responder problem of what genes are important, we can start thinking about our selecting what genes to go after, to edit, as a graph traversal problem. Right, so the idea is we combine the network with the genes that we've learned from our model of which ones are important. And obviously, we're not doing this in a vacuum, so we're going to use some literature as well. And by traversing this network from a couple of starting points, we can construct basically a list of targets, a list of target genes. And these list of target genes, let's say 100 genes, go into gene editing experiments. So at a higher level, how does artificial intelligence help drive these in vitro gene editing experiments, right? Because the problem is that the gene edit search space is combinatorial, right? So we can't, there are maybe 15,000 genes and we can edit one gene or two or three and we can upregulate them or downregulate them. So you can't just naively grid search across it, right? And the solution that we have at Immunai is to use AI to guide a sort of conceptual gradient descent of our in vitro optimization. And what this looks like is we take that list of AI-driven targets that I just talked about, and that becomes our starting point for a high-dimensional optimization, right? So the AI gives good initial conditions, and then we take those, let's say that list of 100 genes, and we go and we run some in vitro cell therapy experiments, right? And those give us the objective function, right? Those give us the sense of how well, for example, after applying each one of these edits in turn, does a T cell kill tumors, right? And then we can then go back to AI and say, okay, given what we just saw about how well each of these edits helps a T cell kill tumors, what's the next set of targets we should go after, right? That maybe hones in on a particular area that we want to focus on. We do this, and the great thing about Immunai is we are sort of AI is really coupled to our in vitro cell therapy experiments. And so we're able to do this multiple times, right? So instead of having to sort of get it right in one shot, you really think about it as sort of walking down a optimization landscape. Of course, the final test, our sort of held out set, so to speak, are animal models initially, and then ultimately clinical trials in humans. So in conclusion, at Immunai, we believe that our understanding of the immune system is at a sort of wonderful historical inflection point. And this is thanks to many things, but in particular, thanks to lots of new technologies just in the last handful of years, especially like single cell multiomics, which is our ability to interrogate and understand what individual immune cells are doing. And combining all of that allows us to paint this incredibly rich picture of what an immune system is doing in the face of a particular disease. But understanding all the data thrown off by these new technologies requires really tight, really multidisciplinary collaboration. It's not something that AI can do by itself, I promise you. And I would love it if that were the case, but it's not. Right? And so when we think about what can we do at Immunai, we're building a world-class data asset to help answer some of these questions and a portfolio of AI methods that will help us learn how to reprogram the immune system. And I think that 10 years from now, 20 years from now, we will be able to modulate the immune system so well that I think it'll turn things like cancer, many types of cancer, from lethal diseases into diseases that can be managed and become more chronic. And I want to be part of that. If what I've talked with you today is exciting, come join us. We're hiring across almost all computational roles, from software engineering to computational biology, senior level data scientists and machine learning. If you are motivated by hard problems, if you are excited by the idea of pushing the boundary of biological and clinical knowledge, if you really enjoy working with people who don't have the same background and experience as you, and if you're sort of temperamentally suited to working at a high growth, very exciting biotech startup, and of course, as a bonus, if you have a strong track record of high quality research publications and or experience with multiomics data, please get in touch with us. So thank you so much and have a good day.